And yeah, I appreciate the invitation by Professor Wang and have the chance to communication with all of you. And yeah, it's my honor to introduce my work recently. Yeah, so let's start. As we know, there are four types of the macromolecules, like nucleases, proteins, lipids, and glycans. The nucleic acid is the fundamental of the life, but the gene cannot explain the whole life. As a conductor of the life, the protein was templated, synthesized by the gene, by the guidance of gene. But most of the time, the protein was exist with various platform. The post-translational modification is one of the reasons caused the protein form increased significantly, especially for the glycosylation modification. Yeah, for the glycans, we know most of them was exist with the glycoconjugates, like glycoproteins, glycolipids, and glycoRNA, which was discovered recently. All these glycoconjugates involved in most of the biological process, like the signal transduction, cell differentiation. It also related to many of the disease, like the viral or bacteria infection, and some for the cancer development and metastasis. As we confront these years, the COVID-19 was spread over the world. The S protein of the virus was hyperglycosylated. So there are 22 glyc sites on the S protein. The site-specific protein glycosylation analysis accelerated the vaccines study. All the site-specific glycoprotein analysis was based on the mass spectrometry. For the analysis, there are three parts. For one is for the glycosite analysis, which can be performed by the glycoproteomics. It's similar to the proteomics. The glycan can be released from the peptides, and we can analyze the deglycosylated peptides. The second part was mentioned by previous professor. The glycan was released from the protein, and it can be native or derivatized to identify by the mass spectrometry and to annotate the composition and the structures. The third is the site-specific glycosylation analysis, which the intact glycopeptide can be directly analyzed by the mass spectrometry. Yeah, this part is very challenging because the intact glycopeptide the abundance is very low for every glycopeptide, and because the isomers of the glycans, they are many heterogeneity, and in the mass spectrometry, the ionization efficiency is very low. In the data analysis of the mass spectrometry, the false positive is really high, and it's hard to be removed. So in our study, we use some of the specific column maps to enrich the glycopeptide and also gonna separate it. We also developed the sample-specific glycosite database to make the false positive decreased. So using this strategy, we can identify more than 10,000 unlinked intact glycopeptides, and which is increased 33 times than the helic enrichment method. And in this method, we also can identify some atypical glycosides in the intact glycopeptides. Normally, for the glycopeptide enrichment, it need take more than 24 hours. So to shorten the sample preparation time, we developed a one-step enrichment method to rapid the sample preparation. In this method, the time can be short to less than 1 hour, and the intact glycopeptide ionolysis was also increased because some of the longer peptides and hydrophobic glycopeptides were identified in this method. Using this method, we applied them to the protein ionolysis, glycosylation ionolysis in the cell and body fluids, tissues, and extracellular vesicles. I will briefly introduce the extracellular vesicles ionolysis. So we know that the EV or MEV is secreted from the cells, which was enriched with bioactive contents. The MEVs was applied in the clinical diagnosis, drug delivery, and some of the therapy. In recent study, the bovine derived milk derived EVs was used for the cancer therapy, but the protein was not identified in the surface of the EVs. So we developed the method to ionize the glycopeptides on the bovine, caprine, porcine, and the human glycopeptides. So this is the characterization of the milk-derived EVs. We can see the morphology and the size distribution of the EVs. From the intact glycopeptides ionolysis, we can see most of the glycoprotein in the MEVs is silated glycoprotein, especially for the caprine, the NU5GC silated glycopeptide was the dominant of the glycopeptide, which may be very important for the therapy of the EVs. In the identified glycoproteins, there are 10 proteins were shared in the 4 species, and all the proteins were related to the immune system. We know the MEV was biogenesis from the cells, and the CD63 is related to the MEV biogenesis, and the protein MFGE8 was related to the endocytosis. So from the results, we can see the CD63 was high glycosylated, and they are similar to the different species. But for the MFGE8, it's very different from different species and different glycosides. So besides the glycosylation ionolysis, we also interested in the glycosylation reconstruction of the in the cells. The cell engineering was activated again because of gene editing. Now the engineered cell was seen as an arsenal of the diagnostic and therapeutic. The glyco-engineered cells will improve the next generation of vaccines and immunotherapies. Yeah, with collaborate with Mori Fujita, Professor Mori Fujita, we developed HEC293 cell, glyco-engineered HEC293 cells. Use this cell, we developed a tool for the protein glycosylation prediction. And we can use this glyco-engineered cell to produce the recombinant glycoproteins. And we also can use these cells to study for the glycosylation process, like the ER quality control. For the ER quality control, we know that the proteins and can be glycosylated and folded and decreased by the regulated by the ER quantity control. So we constructed the three knockout cells, and using to investigate the difference of the protein and the glycoprotein expression, we're using the quantitative glycoproteomics to analyze the proteins. From the results, we can see the ER pathway and the lysosome pathway was the mainly affected by the MOGs and the connexin and the calciculin knockout cells. For the ER's pathway, we can see the NPL and the BIP was upregulated, which means the ER stress was happened in the cells. And for the lysosome part, we can see the HEX-A and GUSB was downregulated. This protein was in the lysosome, and some of the LAMP was upregulated on the membrane of the lysosome, which was different between the lysosome. So to see the difference, we checked the exosome, the secretion of the proteins. We saw that the GUSB and the HEX-A was secreted to the outside of the cell. With the mannose phosphate was decreased in the knockout cell, we can know the GUSB and the HEX-A was not the excitosis was not because of the lysosome. It's because the directly vesicle excitosis. So this is the story, and the last story is about single cell omics. As we know, the single cell omics is a powerful tool, can reveal the heterogeneity of the cell state and the tumor immune microenvironment. As reported, the glycosylation is also related to the cell state, but for the single cell glycomics, it's really challenging to be well developed. To develop this method, we developed the method to analyze the proteins and its glycosylation using this platform. In this platform, we can analyze the peptides, intact glycopeptides and deglycopeptides with a similar strategy. At first, it's a single cell proteomics. From the results, we can see we identified more than 1,000 proteins from a single HEC293 cells. And we use this method in the lung cancer tissue, and it performed very well. And we cluster these cells to 7 types, and we can see in the cancer tissue, some of the immune cell was entered into the cancer tissue. And for the glycosylation, single cell glycosylation analysis, we developed a site-specific single cell glycoproteomics. In this method, we can identify more than 1,000 intact glycopeptide in 100 cells, and about 25 intact glycopeptides from one cell. And for the site-specific glycoside analysis, we use the endoglycosidase cleavage the glycan from the peptides, and we see there are more than 230 glycoside was identified in our study. So that's all for my presentation. At last, I acknowledge all my collaborator and colleagues. Thanks for their support, and thanks for all your attention. Thanks.
工程创造人类美好未来。